From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 59, recorded on August 26th, 2020. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Good to be with you from LD Nursery Studios, my new perch here for podcasting.
You got yourself a little teapot or something there, right?
Teapot, and I think there's a little sleeping bear in the background, so
yeah,
it's legit.
Nice. And I see your mixed pre-recorder there.
Oh yeah, we're running, so it's recording, and
nice.
We're up and doing it.
How are things in Salt Lake? You got cases these days?
We do. So continues to. There's a little bit of a downtick, thankfully. Although we're still hovering in the, I think it's in and around 400 or 500 cases a day.
Mm-hmm.
Of course, everyone is sort of holding their breath right now with the opening of the universities, colleges, and universities.
Yeah.
And, obviously the early returns, places like North Carolina, I saw Notre Dame, Alabama. Oh my goodness. This is, feels like things could break in a not good direction here. And so we're wondering about that as well.
What are the plans in your university for classes?
So it's a hybrid model, a lot online, but a few things in person. Certainly, small number lab classes, for example, where almost the format dictates that you would be together. But they have, I think, to their credit, built in some flexibility here and announced that up front that all bets could be off. And in fact, I think if you look on the headlines of the local newspaper today, some of the residents' assistants, the dorms, are threatening a strike. As they're seeing the numbers and maybe the disconnect in some ways between what we hope will happen and what will actually happen. How about in your neck of the woods? Similar situation with school opening up, or what are you seeing?
Well, Columbia is all online this semester.
Yep.
Only a limited number of students are allowed to come back to the dorms, just those students who didn't have internet access, basically. And they think it might even go on next semester as well. We'll see. The medical school, same thing. First semester is all virtual. So that's going to be interesting. And I mean, I only have one person in my lab, Amy. So she's been going in. She started the SARS-CoV-2 project, so she was going in every day, basically. And I'm going in 2 days a week, just to minimize exposure,
Yeah, you and me both. I'm a similar schedule. And then we're doing, I can't remember if I mentioned it on our previous episodes, but we're doing pairs of people at 4-hour shifts.
Yeah.
And we never change the pairs. So we're trying to minimize the interactions, right? It's always the same partner in a different room.
Right.
It's one person per room, per shift, and everything like that. But yeah, crazy times. And it's going to continue, I think, for several months at the least, or more.
Yeah, for sure. Well, it's very interesting. Some countries have very low infection rates, like Italy and France, the UK, Germany. They're all very low. So we'll see as schools open what happens. I mean, my prediction is that cases are going to go up, right?
I think that's already happening. I think I just saw a headline saying that the University of Alabama has more cases yesterday than the country of Canada, for example. And so a pretty sharp contrast in how we've approached this as a country.
I mean, without a testing plan, you can't open really very effectively.
Yeah.
It's not a good idea. Did you see today or yesterday the CDC said we're not going to test people who are not symptomatic anymore?
Yeah, this sounds like we're moving backwards.
This is crazy. This is totally crazy.
Exactly. And at a time, too, when there are some, wrinkles of hope. So I think the advancement of saliva testing instead of the little pharyngeal swabs that are pretty invasive, that got deployed, at least so far, somewhat effectively by the NBA.
Yeah.
I mean, if we compare professional sports teams, where have things kind of been, at least in the short term, viable and sustainable? And, I think a few universities really deploying that in a more organized and comprehensive way where you could imagine doing better. And yet, yeah, that I saw that CDC news and the head scratcher, to be generous, and just malpractice, I think, to be a bit more accurate about it. Yeah.
Such is the
Well,
case.
Vincent, we're, oh, actually, I wanted to ask you. So, last time someone noted or asked or told me that you actually posted us on YouTube. Are we doing that again? Fantastic. That's a relief because I spent about an hour in hair and makeup before.
Yeah, I mean, it's hard. It's tough. I know. Yeah, that was the first one last time, I think.
Yeah, pretty cool.
With Simon and Heather. And yeah, I put it up because I figure I had it there. People like to see, so there you go.
Yeah, no, I think it's a great idea. And speaking of that, last time you were in the sort of podcasting nerve center in the basement of your house.
Yeah, that's right.
Guitar lighting, blue lighting on your guitar in the background. It looks like you have a different background today. Where are you coming from?
Yeah, I'm in my, we have a little summer house here in New Jersey. And I'm sitting up against the wall because this thing here is a circuit breaker box. It's really exciting. Yeah, there you go. See that little hand? I should take it away. It doesn't look very good. Well, I can't. Yeah, I'm down here, but as my wife says, you're just doing the same thing you do all the time.
Working vacation, but at least you can get some fresh air, I'm guessing, and some.
Yeah, it's nice out. I mean, we have a sailboat. My son wanted to sail, so he's actually working down here for the summer. And he wanted to sail, but he works during the day, and so we haven't been able to get out yet. We put the mast up on Saturday, and we haven't done anything. So.
Well, hopefully this afternoon, or maybe in the next.
Well,
day
now it's 4:00. Too late now, I think.
It might be dying down a little bit.
Although it is pretty windy because there's some storm coming here. There's some kind of hurricane in a couple days. Although that shouldn't be doing it. It's pretty breezy. You could go, but I'll see. He usually doesn't want to go.
Yeah.
But yeah, it's nice air, and the ocean is just 5 minutes up the road there, so it's kind of nice. But I set up a little studio here. I got a desk and my computer and recorders and everything, and I'm here. I can do this anywhere, I guess.
I love it. That's fantastic.
Yeah.
Well,
I did a TWiEVO today. I did an immune on Monday, and Friday we have another TWiEVO, and then I have a thesis committee meeting tomorrow. So I guess I'm crazy, basically. But
working vacation, I think, is the term. But
I don't know. I'm okay. I like it. I kind of like it. I was watching something on some video. My wife was watching. I don't know. It said Wasatch, and she said, "Where is that?" I said, "It's got to be Utah, right?"
Yeah, Wasatch Front, the westernmost of the Rocky Mountains.
It's near you.
It is. I'm sitting on it in the foothills.
Okay.
Yeah.
I forgot what it was about. It was pretty interesting. That's the only thing I knew. I said one thing I'd learned from Nels: Wasatch Mountains are right there in Utah.
It's starting to rub off.
How's the kid? How's the kid, Nels?
It is good. Thanks for asking. 10 weeks, 2.5 months. So the nursery is still a podcasting studio. This might be my last recording. I might go back to LD Lab Studios next time. We'll see. Stay tuned on that one. But no, we're having a blast. She's doing well, growing fast, and sleeping even some. So we'll take it.
Before you know it, she'll be in college.
I can see how it could feel both, especially the early days, sort of the hands-on intensive days can feel both sort of, all consuming, but also just go by somehow in the blink of an eyelash. So, yeah.
So I have, my son went to Marist, my younger son.
Mm-hmm.
It's a computer science major. He didn't finish one course he needed to graduate. He finished 4 years. So this fall, he's got to take one. Of course, he doesn't have a dorm. He has nowhere to stay. So he wanted to take it online. And the professor said, you have to come 1 day a week to class. Wow. The rest you could do online. So he's going to do. He's going to drive up and back because it's only 2 hours, but he had to get tested.
Oh yeah, of course. Yeah.
So
That's amazing. Complicated.
we sent him up there, and he got tested. We're waiting to hear. I'm sure he'll be negative, but if you're positive, you can't go to class. But they have classes. It's up to the professor actually what they want to do, but many professors are having classes. So
Wow. Yep. It's complicated. A lot of institutions sort of bending in pretzels to either. I think we're doing a hybrid approach here.
yeah,
Whether that sustains, I think, is a big question. Some of the early returns on universities that have tried to do in-person stuff has been pretty dismal, to be generous. And
yeah,
so,
for sure. Oh, I think as soon as you open up, you're going to have outbreaks, and maybe you have to shut down. I don't know.
yeah, we'll
I mean,
see.
I hope I'm wrong. I hope I'm wrong, really do. But it seems to me that you bring in a bunch of kids who are infected, and you're going to get more transmission. So,
Of course, yeah. And I have some friends with college-age kids who decided to take a gap year, and I think I give them a lot of credit for
yes,
looking ahead, sort of simplifying, looking at the
yeah,
little
it's
bit
a
of
good idea.
Yep.
I have a few people who have written me saying they're taking a gap year. Yeah, it's a good. They got in, but they said, "Let me delay." I think that's very smart because it's just too crazy. Online is not great; it's not perfect, and might as well wait a year. Good idea.
I think so. And to kind of, even just reassess that notion of what it means to, this idea of falling behind or keeping
Yeah,
up, which is a pretty arbitrary definition. I mean, I think in our careers in academics, we could certainly, point out years of space that we wouldn't trade for anything, that you could look from a very strict academic definition and say, oh, that was a gap year or a year that weren't pursuing education in a formal way somehow. And yet, if anything, I think a lot of our colleagues would look back and say those were the years that were just as important as the formal education. So,
sure.
Rebalancing the equation a little.
Yeah, you bet.
Yeah. Well, anyway, so Vincent, we're coming up on a big milestone. Next episode will be our 5th year of TWiEVO podcasting.
Look at that, 2015, huh?
Yeah, 2015 to 2020, episode 60, once a month for 5 years straight. So pretty excited about that. And stay tuned. We have some sort of special ideas in the works. We'll see if we can pull this one off next month. In the meantime, we've been having this summer has been, I think, 2 episodes. We've been a little slow on the episodes, but 2 great episodes with some guests in person. And of course, our conversations have sort of spilled over longer than our, kind of up to the podcast limit or beyond. And so we've skimped a little bit in responding to some of the great mail that we've been receiving. And so for today's episode, we're going to tie up some loose ends. We're going to read some of that mail, respond. And then we even have a loose end paper. This was, I don't know if you remember, but right when the pandemic was coming on, we were podcasting as usual. And it even had a virus evolution story that I've been threatening to return to. This is my hope for today, is that since we'll just move through the mail, that we'll have a few minutes so we can, at least just put this guy on the table, tie up some loose threads, and sort of clear the decks so we can charge into our 6th year of podcasting, which I'm also looking forward to.
All right. Let's see here. We got a couple. Why don't you take that first one?
Will do. So the first email is from Caleb. He writes, "Greetings, Drs. Racchianello and LD. I first and foremost would like to thank you for the endless entertainment and education over the last 4.5 years. I remember where I was on the first episode of TWiEVO when it went live. I was pulling into a parking lot heading into lab, and how excited I was to listen. The podcast has been with me since my PhD began, September 2015, and has inspired me to explore fascinating evolutionary questions. The first chapter of my dissertation has just been published, see attached, and I wanted to share it with you to show you how impactful your podcast truly is. I work in a veterinary entomology lab where the majority of the research conducted is field-based, working on dairy farms and at poultry facilities, to understand some aspect of an insect or arthropod biology to develop improved methods to control them. My dissertation project strays far from the standard scope of what my PI has traditionally done. I wanted to figure out what was causing a strange form of insecticide resistance known as behavioral resistance in the housefly. Without writing out my entire dissertation, and thank you, Caleb, for a little editing here, behavioral resistance is the heightened ability of an organism to detect and limit contact with a toxic substance, perhaps as a result of a repellent or an irritant property of the toxic substance, its formulation, or presentation leading to an adverse response. While behavioral resistance has been documented for more than 50 years in the housefly, no one has taken it one step further to try to understand the underlying mechanisms conferring this resistance. The first chapter of my dissertation describes an old school selective breeding technique in which we breed flies to exhibit this behavioral resistance phenotype. Then we do a suite of behavioral observation assays to characterize the resistance further. This experiment, which took over 3.5 years of selective breeding, set me up to have the ability to do some exciting genetic and genomics experiments. I have now completed the first autosomal linkage analysis of behavioral resistance to insecticide in the housefly, which allows me to identify the chromosomal resistance factors where they are. And I'm now collaborating with Jason Stayich, I believe Nels may know him, I do, colleague, on a pool-seq project, essentially an evolve and resequence, in which we sequenced both susceptible and behaviorally resistant flies to look for genomic changes that may be conferring the resistance. My dissertation would, in all seriousness, not be the same without your podcast. I drew inspiration from each episode to continue to push forward to better understand the biology behind what makes us tick. Thank you again for all you do. Cheers. This is from Caleb, PhD candidate at University of California, Riverside.
I just put a link in. I had forgotten to do it, so you can take a look at it.
Oh, this is, yes. So let's link this onto the, we'll put this on the TWiEVO episode page as well.
So this is selection, reversion, and characterization of housefly behavioral resistance to the insecticide imidacloprid. Mm-hmm. Cool.
Very cool, Caleb. And I can't think of a better letter to kick off our opening the mailbag. Very kind of you to say, and glad that we can be sort of, distant sort of science buddies here in the process. That's inspiring for me and inspirational to hear that.
Yeah, it's great that we inspired somebody, Caleb and others, hopefully. It's good to hear.
I know why we do it. And of course, I'm a massive fan of these sort of experimental evolutionary courses, the evolve and resequence idea. And so here, I think Caleb explained it in his letter, but to kind of fill in a little bit of the details, you can do this under many conditions. We do this in my own lab with vaccinia virus, the vaccine strain pox virus that was used to eradicate smallpox. And with the notion being you can start, because you can get rapid evolution. We've talked about this on TWiEVO, whether it's yeast, a virus, in this case, houseflies. You can, because the population size, the generation times are work in your favor, you can actually see adaptations in real time before your eyes, in this case, 3.5 years. In some cases, 3.5 months, depending on how you set up your experiments. And then the ability to sort of, best case scenario, freeze down the parents or the ancestral strain at the beginning of your experiment, and then have the ability to, in some cases, compete these directly, ancestor to adapted sort of live version, and then sequence everything, and then ask what is the genetic basis of the differences, the adaptive course that you observe in the laboratory. Yeah.
It's good stuff.
Yeah, really fun.
Keep it up.
Yeah, thanks, Caleb. I'm looking forward to the next chapters as you're closing out your PhD and moving on. Keep us posted on what you're up to next.
Next one is from Per, who writes, Dear TWiVers and or TWiEVOs, I'm just a protein chemist who has started working on virus-host protein-protein interactions. In the beginning of January, I got a tip from a virologist at my department to listen to TWiV, and I have since been a devoted follower. You have really helped me to better understand the virology field, how virologists reason, and for this I'm grateful. Many thanks. I've also ventured into protein evolution without a formal education in evolution, so I don't know if this is a stupid question, but I take the risk. It's clear from your discussions that SARS-CoV-2 has not evolved to be more transmissible because there's no need i.e. it wouldn't provide a selective advantage. But with all lockdowns and physical distancing, the R value has dropped in many places around the world, thus making it more difficult for the virus to spread and possibly imposing a selection pressure for increased transmissibility. When I had thought about this for a while, I realized that this must happen to most viruses when there is an increased immunity in the host population after vaccination or after the virus has swept through and infected a large part of the population. Is there evidence that any virus has evolved within a host to become more transmissible due to fewer available hosts, which to a non-expert like me appears to represent a very strong selection pressure? If not, this must be exceptionally difficult and could perhaps be used as another argument that viruses rarely or never evolve to become, quote, worse, unquote. Or would such evolution be rare because the window is too small between high enough access to new hosts on one hand and too few hosts to proliferate at all on the other? Am I thinking correctly about this, as Rich would say? P.S. I try to be concise despite writing from Sweden, and Per is at Uppsala University in Uppsala, of course.
Yeah.
Well, it's tough to know what's enough transmission for a virus, right? Because we're looking at it from our human viewpoint, and we're thinking, oh, maybe 100 hosts is not enough. Maybe it needs millions, but who knows? Who knows what is enough, right? And so I think it's a very hard question to know if you're quarantining and distancing and lockdown and all that. Is that going to make a difference? Because I think the infections are still spreading because there are always people who are transmitting. And really, in my view, you only need to find one more host and then one more after that and then another, right? It's enough. You don't need to find thousands and millions. So I've learned to try and back off against these humanized ways or these human values, I should say, to put on biological properties because we simply don't know what they are. Now, have we ever seen a host and a virus evolve when there's not many hosts around? I don't think we have ever made that observation. Maybe not even experimentally. We don't do these transmission experiments experimentally. And it would only be in the nature. And most hosts are ubiquitous, right? They're plentiful. So what do you think it's ever been done, Nels?
Yeah, well, it's a couple of points. And actually, first to get just to the beginning of Per's letter, actually, just a protein biochemist, a very Scandinavian, modest opening. I'm just an evolutionary geneticist over here myself. But no, I think I think you are thinking about it in a productive way, which is to provoke some thought and think about it. And so, one thing, and I agree with you completely, Vincent, about sort of trying to back out and look outside of our human eyes or our kind of anthropomorphic view of things. And so, I guess a couple of points along that line. So one I would think about is when we're thinking about transmission is maybe less about how many hosts there are, or how the virus to virus competition actually, regardless of how many hosts you have, and how much competition is there for that resource, basically. And we don't talk about this as much, but so are the virus, and I think what ties in hand with this is also then the action of selection. And this is really complicated. So this has been, we've talked about this. This has been a massive topic on TWiEVO. And by the way, Per, I think you picked up on TWiEVO in January, right when things really heated up here with the pandemic. This has been an incredible multidisciplinary course, all the way from the virology to the evolution to the public health issues. And so again, our listeners, this has been a ongoing online course basically around SARS-2. Really worth it to hear some of the experts and voices there. But so if we think about, and this is really difficult because so we've talked about this point of how do we distinguish between selection and then just sort of demographic population genetic randomness. So in particular, we focused a lot on this, is it the D614G mutation, which still I think is bubbling up in the news and is sort of controversial as to whether this is making the virus quote unquote worse or more transmissible or something. And in a lot of ways, the jury is still out. So certainly that mutation to D to G, which is the amino acid letter, the shorthand for that one point mutation, that one change in the DNA in the RNA code. So that the version that has the G encoded there has certainly come to dominate a lot of the globe. And so it's still there's a few phenotypes in terms of, in the laboratory mostly, and on Petri dishes almost exclusively, that might speak to it having the ability to, for selection to see a difference in function as it relates to this. But still, you not definitively ruled out. And so when we think, though, about this notion of increased transmission, I think that would be one way that I would consider framing it, is if it's through a selective prospect. Is one virus competing another to the detriment of the other, would be one way of framing it. In terms of examples, so I agree with you again, Vincent, that experimentally, very early days at best. So if we then turn to some natural examples, I can think of two off the top of my head. One is, and this kind of goes to my biases in pox viruses. So the myxoma virus, this is a virus of rabbits that was released purposefully in Australia, and really wiped out the rabbits. And so the number of hosts declined tremendously. In the case of SARS-2, there's plenty of hosts. We've been, for all the ups and downs and the issues in our country in particular, ongoing issues, there's still a lot of naive hosts out there. In this 1950s sort of natural experiment or biological control intervention, for a while, this was working. So I think 90+% of the rabbit population was decimated by this virus. And so here we actually we can peek into, and this work is ongoing. So there's still freezers full of virus strains that were collected in, I think, the 50s and 60s. Eddie Holmes and some others have been looking at this and published some papers along the way. But it kind of pulls back the curtain both on the host side of some of this evolution and on the virus side. And what you end up seeing, and sort of the take home is, guess what? The rabbits are back. So there are rabbits that evolved resistance to the virus. Or another way of saying it is, there could have been existing rabbits that had resistance in the starting population that were selected very strongly. These were the ones that lived. And then in the meantime, you also see a really strong signal of virus attenuation. The virus has become less deadly over that time. Now, I can't recall off the top of my head how much it was looked at, if anything, on transmission itself. This thing was already transmitting really well, kind of similar, I think, to the case with SARS-2, where this doesn't seem like that is much of a bottleneck. The ability of the virus to move to hosts is out there. The others, so anyway, lots of open questions, but that would be one place to look to sort of see something like a approximating experiment with some of these dynamics. Okay. The other case, I don't know, did you see this paper, Vincent, that just came up a few weeks back? It's sort of this archaeological virology. So some old Vikings were I think 1,000 Viking corpses, or almost not quite mummies, but preserved remains were sampled, and smallpox was recovered. Variola virus, the cause of evasion of smallpox, was recovered. And then doing some of the ancient DNA work, some genomes were at least partially assembled. Kind of a splashy paper. I can't remember. I think in Science about a month or two ago.
Yeah. That's right.
Yeah. And there, so I have to confess, I haven't had a chance to really dig into the data. From the skim of it that I made, a couple of quick conclusions. So one was that, in some of these ancient sampled strains, there were some genes that were interrupted or had been lost. And this is a very common theme of evolution, including evolutionary virology. Sort of less is more. The longer you spend time replicating in a specific host, you become more specific. The virus or the organism, the microbe, whatever it is, will start to get streamlined. It will lose sort of the extra features that might be a drag on replication, copying the genome, et cetera. And so there may be a story of that here, where some genes were lost. I guess the wrinkle on it that they are trying to propose with their evidence is that then if you look back at the modern strains, modern meaning up to about 1970, before the virus was eradicated, you actually see cases where those genes are intact. And so the notion would be that those samples were sort of an off-ramp for evolution. These are the, those were the strains that didn't persist, that there were other ones that made it through and actually held on to those genes or didn't lose them through mutation in selection. And so they try to mount a case actually that there could be evidence that the virus got worse because it almost appears like it gained back genes, although ancestrally it would have those genes to begin with. And so I'm a little skeptical about that. Has a virus gotten worse? So that argument, it was proposed, at least in a perspective piece for that paper. It's worth it maybe to dig in and see for yourself and maybe see if you buy the arguments or the speculation. Those are the two examples that immediately come to mind.
Yeah, so this paper, they got variola from ancient Vikings, basically. They got 13 sequences from 13 northern Europeans, including 11 dating to 600 1050 CE, which overlaps the Viking Age, and they got near-complete genomes from 4 of them. So they predate the earliest confirmed smallpox cases by 1,000 years. That's a long time.
Yeah, really cool.
Pan-European presence of smallpox from the late 6th century, so a little bit earlier than we thought. That's pretty cool.
Yeah, I really like that part, sort of the evolutionary archaeology or paleovirology, I think is the great term that's been coined for this, to actually unearth literally these ancient viruses and be able to assemble the genomes. Now, I would just raise a quick question of how much did we learn about the virology here? That's so I think technically it's sort of like this really cool example of pushing back the timeline. Whether we really kind of had a breakthrough there in terms of our knowledge of virus evolution, I need to, before I would weigh too heavily an opinion, I think I need to read it more closely.
Yeah, I think what it does, though, is just stimulate some hypotheses, not necessarily advancing our thinking, but making us think about some different scenarios, which is always good, right?
Absolutely. And actually, that kind of came to the surface in our last episode with Simon, Anthony, and Heather working in his lab. Just how when you add a few new genomes or new sequences, in that case from SARS-2, or coronaviruses closely related to SARS-2, I should say, it gives you this new context or this new view. And so absolutely adding more knowledge, in this case, kind of this very high degree of difficulty, new knowledge to go back and assemble these ancient genomes. Yeah, absolutely. Very cool.
All right, you're next.
All right, let's go on to the next one. This is Sue Ellen writing, I am not a virologist or any kind of PhD scientist, just a retired Oracle database administrator. There's that word again, that qualifier, just. I mean, if we were listening to a podcast right now, Vincent, about Oracle database administration, I guess we would just be virologists or something like that. But
well, the just is an arc in TWiEVO. People, when they know now to write in, I'm just a doctor, I'm just an English professor, I'm just an Oracle database administrator.
That's right. Okay. But I want to weigh in on SARS-CoV-2 and the idea that it was created in a lab or escaped from a lab, this controversy. So my answer to people who make that suggestion is to look at the evidence that was uncovered when the virus first surfaced. In the first days of the virus, it was thought it originated at a wet market in Wuhan. Why? Because the first 4 people who showed up at the hospital had all been at that market recently. I figure if the virus started at the Wuhan lab, then the first group of people who showed up sick would have had some connection to the lab. Maybe not employees, but certainly family members, associates, neighbors. Excuse me. After all, the point of the investigation is to find what the infected people have in common. Do they live near each other, work together, have a common associate who is also infected? That's how it came from the fish market origin story started in the first place. And as for having it been engineered in a lab, well, I just had to had a discussion, read a heated argument with a friend, maybe a former friend, about that too. So let's just say I'm a mad scientist, I told my friend, and I either want to intentionally kill people with a virus, or I just want to find out if changing a gene here and there can make it more virulent or more transmissible. What virus would I start with? Probably SARS-1, which is pretty darn transmissible and virulent. I would probably not start with a novel coronavirus that I had just discovered in a cave, wet market, or wherever. And I don't even know the virulence or the transmissibility of. Actually, from what I've learned on TWiEVO and TWiEVO, I would probably not pick a coronavirus at all, since they have that proofreading thingy, scientifically speaking, that would make my work harder. I'd probably use the flu virus, since I could make mutations easily, and that's a pretty darn good spreader. So I'm able to make better arguments thanks to the Twix podcast. I make fewer friends, but that's another story. You're preaching to the choir there, Sue Ellen. And one quick non-coronavirus question. Is cowpox still a thing? Like, does it even still exist? You were all talking on TWiEVO recently about smallpox in terms of vaccination, and it got me wondering. It seems that in the olden days, milkmaids and others who worked with cows got cowpox on occasion, but I have not heard of a case of cowpox in my lifetime. So wondering if it has disappeared. Maybe the smallpox vaccine took care of cowpox too. Just curious. Keep up the great work, guys. Love the shows, all of them. Sue Ellen in Roswell, Georgia, where it's cold and hot and then hot and cold again. Yesterday's high 65. Today will be close to 80. And our state is open, but really and truly, most of us are still in our bunkers. We'll let the crazy rednecks test out the transmissibility and virulence of the virus for us. A lot of ground to cover here, Sue Ellen. A great provocative letter. So maybe, and I thought there was a really, let's cover the origin story, both in terms of the where did SARS-2 potentially emerge from? Very much an ongoing and open question. And then also that notion that it could be lab created. And so I'm sure you saw this, Vincent. There's sort of two articles, I think, just sort of commentary pieces that showed up in Science maybe in the last month or two. The first one was a science reporter, I think, just trying to kind of explore both questions with some investigative reporting. And again, this is, massively complicated, high degree of difficulty stuff because it's become embroiled in kind of global politics, to say the least. And then a accompanying piece or a knock-on piece from the person, and I'm blanking on her name, I'm sure you remember, who runs that lab at Wuhan, who's been there's been some accusations from some officials in the Department of State, in particular in the U.S., about sort of throwing fuel on the fire, at least to try to suggest that there could be something going on beneath the surface here. One of the striking things from that commentary from
Xiangli.
Yeah, Xiangli, was that she pointed out a couple of points that Sue Ellen raises. So first of all, there hasn't been a single person from her lab or that institution that has had a SARS-2 case, which again connects it to that, just as Sue Ellen is saying, sort of following the leads here and thinking about what's in common. And then secondly, she pointed out that they actually only have, I think, 3 strains total that can replicate in the lab, and none of those 3 are anywhere close to SARS-2. The rest of the sampling is all sequences, right? So this is, go out, right? You recover nucleic acids from bats, and then you assemble a genome, but you actually don't have a working replicating virus.
Yeah, right.
And so, of course, you could say, oh, well, of course she's going to say that because that's sort of in her fit. But I think I would take her word for it. This is someone, there's more people talking and framing the issue than just her. But this is a good colleague with a track record over decades of work in this area. And so to immediately go kind of down the rabbit hole of closer to conspiracy theories about lab-generated scenarios, et cetera. And so then, and Sue Ellen, you kind of pivot into this space. And this is, I found myself doing this actually at a poster session for some years back when I just very first started working on pox viruses. And someone came up and I was talking about actually experimental evolution, right? And so to adapt the virus to a certain condition. And so someone came who has a very strong interest in biosafety and immediately kind of castigated me for what he was saying. Oh, these are gain of function experiments. You're going to create a super virus, and so convince me that I should trust you. kind of a, I think a little bit of a shady approach for a poster session with some really preliminary data. And I almost caught myself. So I said, oh, well, if I wanted to be a bioterrorist, here's what I would do. And that kind of went a little bit down I caught myself. I don't know if I would like actually win over any confidence from someone asking these questions if I sort of said, oh yeah, there's 10 easier ways to launch something like this, right?
Yeah.
So
I'm not sure that helps. No.
Yeah, exactly. It doesn't necessarily help your
Yeah.
cause, depending on what your or win you friends, depending on what your goal is. And maybe it is to have a good provocative argument, and that's okay too. But no, in terms of some of and I think this is important for our TWiEVO listeners, and especially as Caleb, our last letter was describing, how do we think about these adapt and resequence experiments, especially if we're talking about infectious microbes. And so there are some common sense things you can do to think about how do you sort of include biosafety. Experiments we often start with very attenuated viruses and then ask them to improve only towards the quote unquote wild type. And so we're sort of trying to recover to what is already known for a highly attenuated strain in the first place. And then the other thing, and I think is a really important point, is that, what are the most dangerous things happening in terms of superbugs? And that is in the wild. These new collisions with bats and villagers in China or other locations around the world, where for the very first time, viruses that are replicating under the full complement of all of the biology between potential spillover events is being tested day in and day out invisibly. We almost ignore it because we don't see it. And what we do see are some of the virologists who are doing, in comparison, a very modest number of experiments in controlled lab settings that don't sort of rage out of control. And so I think, this has been a point of discussion as well, but the real energy in terms of, and even when I think of bioterrorism, I mean, my first thought goes to what are all the things I can't see that these viruses are doing in nature. And in fact, that's what we're up against right now, right? Wherever this emerged from in China and the exact origin story of it, which is many labs pursuing, and with some political wrinkles thrown in. But I think that's where putting our energy, our eyes, our resources will really help us, hopefully, to catch the next one before we get to a situation where we find ourselves today.
I think for me, the best argument is that all the data we have points to SARS-CoV-2 originating from bats. There's a lot of data, including observations that it's recombinant from different viruses. You can see where all the pieces have come from pretty much and trace how long they've been circulating. Whereas all the alternatives are accusations that have no data to support them. Oh yeah, she had it in her lab. So where's the data? You don't have any. It's just an accusation. So as a scientist, I look at the data. That's it in my opinion. Yeah.
So then Sue Ellen also has a non-coronavirus question. Is cowpox still a thing? So it turns out it is. So it's there's not a lot of it out there, but it's circulating in rodents. This is an important, I think in Europe and maybe in South America, certainly in Europe, kind of percolating beneath the surface. And, this raises a few important points. First is that it's called cowpox, but the host range is pretty different. This is a key for virology and evolutionary virology in particular. Sometimes our names fall very short of describing what's really going on with the biology and the range of these things, or even the reservoir hosts, which might be sort of the key place for thinking about where these things are coming from, replicating like crazy, adapting, and then potentially spilling over in some rare cases. So cowpox is pretty interesting. So it's kind of a super tanker among the pox viruses, at least the well-known ones, meaning that it's a very large genome. It has, I think, about 250 genes, whereas, for example, variola virus, smallpox that causes smallpox, has more like 190 genes. And for every gene, you can like shave or add a KB to the genome as a rough thumb rule for pox viruses. So cowpox was used originally in some very sloppy vaccination cases. There's still some controversy about whether it was cowpox used or horsepox, which is another sort of slippery slope of naming and virus evolution. A really fun story, though, if it really truly was a horse virus, then instead of vaccination, which is the origin of the word vaca, meaning cow, we should be talking about not vaccinating ourselves, but equinating ourselves, the equine version from a horse. But that's kind of a side point. Is sensitive. These are close enough relatives among the so-called orthopox viruses that you could stop it very quickly with a vaccine. And in fact, there's really no human cases to speak of. There's a different pox virus that has been proposed as it's a select agent and has been proposed as one that could emerge from the wild, which is monkeypox. Again, take that name with a grain of salt. But this is spilled over in a few cases, actually in pet store scenarios, I think, again, through rodents, not through monkeys. And so whether or not this has the same potential as, say, coronaviruses or some of the other emerging pandemics, probably we're in a slightly better position in terms of the success with vaccinia and the experience or history with these sort of live attenuated vaccines, and even some antivirals that have been stockpiled to like great cost and resources, thinking about a potential smallpox sort of bioterrorism situation, hypothetically. Anyway, yeah, just to hopefully put in some answers to some of those great and curious questions.
All right. Xiaoyu writes, "Hi, Vincent and Nels. I'm a big fan of TWiEVO. Probably Nels still remembers me. He was on my thesis committee 4 years ago." You remember, Nels?
Of course, I'm not on that many thesis committees. And Xiaoyu is one of the most talented students who came through our program. And so I'll say a little bit more about that. But of course, I remember.
I went to college in Wuhan, and my wife is from Wuhan, so this pandemic feels very personal to me. I'm very tired of the baseless Wuhan Institute of Virology origin theory. The U.S. origin theory floating around in China is even more ridiculous, and most scientists rejected that because there's no evidence. As someone who knows the city pretty well, here's my argument that it was not from the Wuhan Institute of Virology. Lab pathogen leaks occurred before in China. Back in 2004, there was a SARS lab leak in Beijing, the original SARS, and the whole lab was sent to quarantine hospital. Even last year, there was a Brucella outbreak in Lanzhou. In both cases, grad students and staff were infected. Most lab leaks are like that. They start from people working in the lab to others. This time, no one from the Wuhan Institute was infected till February 2020. I think this is strong evidence suggesting SARS-CoV-2 did not originate from the Institute. And that's exactly what Sue Ellen had suggested as well. Mm-hmm. If you don't believe the words from the Wuhan Institute, the spread pattern of COVID-19 in Wuhan City did not support the Institute origin. Wuhan is a big city with 11 million population, separated by the Yangtze River, the biggest river in China. The Institute's in the south, while the wildlife market most of early cases and the hospital first report the disease in the north. Many hospitals in the north were overwhelmed by fever patients in December 2019, while the big hospital close to the Institute, Zhongnan Hospital, was still remodeling its ICU, preparing for the outbreak on January 6th, according to published interviews in Chinese. I also have doubt about the early patients. Remember, no one knew this virus. There was no test at the time. I don't think we can rule out that these early cases started with a bad flu, stayed in hospital for treatment, and subsequently were infected with SARS-CoV-2. Unless the sample was taken in early December, which is rare for flu patients, I'm skeptical of them. I still think the wildlife market is the center of this spillover. Please let me know what you think. Xiaoyu is a postdoc in the Wang Lab at Washington University in St. Louis. So Xiaoyu, I think your arguments are good. I wonder why you think it's I guess by the wildlife market, you mean the fish market, because, the very first case now is not even associated with the fish market. So it was my understanding that people are no longer thinking that was the origin of it.
Yeah, I agree. And it's that difference between, like where the first dust up happens in terms of kind of clinical contact, but then still tracing it back from there. And I think that's pretty common kind of run-of-the-mill epidemiology that, getting back to patient zero is a pretty massive challenge, whereas patient, I guess, hypothetical, whatever, 10, 20, 100, whatever it is, that's kind of where you first see it in the sort of outburst. But I do want to just step back and first just say, Xiaoyu, great to hear from you. And Xiaoyu was in Cedric Fachat's lab for his PhD. Cedric came from University of Texas, Arlington, had his lab at Utah for many years, and Xiaoyu was doing absolutely beautiful work thinking about the retrotransposons, in some cases of bats, and really creating new knowledge around this. Not only a spectacular scientist, but also one of the funniest guys I've come across. Amazing sense of humor, really just added tremendous life to our floor over the years that he was doing his PhD training. So anyway, fun to remember a little bit of that. And then, yeah, a really personal connection, given this is where you're from. And I think that's been missing largely from the conversation, where, we might hear some whatever news piece or provocative thing that's leaked to the news through the State Department or some kind of, it just doesn't, I think getting back to these real conversations by people who know the geography of the city, who know some of the details. And then, so certainly, and I'm not trying to mount let's just be naive and, just believe what everyone says, but to follow real evidence as we can gather it from diverse sources and to weigh that evidence in a reasonable way. I think this is how we move forward as best we can. And that's, I think it's happening, but it's just sort of more among quieter voices. And so what we hear are those people yelling or trying to gain attention for political reasons across the board and in different sort of scenarios. And that's really been, unfortunately, a massive distraction, which has also led to some absolutely horrific public health decisions ongoing in some cases, as we were kind of discussing at the top, and has really slowed down our ability or, thrown cold water on some multinational institutions we depend on, the World Health Organization. And as we go down these sort of conspiracy rabbit holes, everything gets worse instead of better. Whereas if we weigh the evidence, listen to sources that aren't sort of, kind of super political, that we can have a way of actually stepping through this in a more positive way. Okay, so our next letter is from David. He writes, "Dear Chronicles of the Progress or Progression. Here in Western Massachusetts, we've had 3 days in a row with temperatures above 29.5 on the centigrade scale. People are complaining bitterly. When I see that Phoenix hasn't seen a low below 29.5 for 2 weeks, I figure I'll just suck it up." Okay, in TWiEVO 57, Jesse says that he is interested in getting more into the evolutionary side of things. And I was reminded of that combinatorial biologist that you had, sorry, his name escapes me, who essentially said that people who are trying to draw a line from pangolins to bats, where there are hundreds of coronavirus variants, and somehow by an extraordinary coincidence commonly found in fairy tales, that is the one that gets into the human, those people are missing the point. It all comes from bats. Bats have been shedding coronavirus into the entire mammalian population through deep time. When we ethnocentrically, you know what I'm trying to say, decide that this new one that is infecting humans is interesting, we will invariably find a coronavirus in some other animal that is the closest match yet, until we find another animal with even a closer match. Because bats have been infecting animals since forever, and there will always be an animal with the next closest match. What I find puzzling is, why is this happening in Chinese bats? Do they have a greater overlap or interaction with humans than bats in other regions? Are the bats endemic to China especially good at bat to another mammal transition? Would bats in Seattle be good enough? Would crawling through caves and scraping bat guano off his hands and knees be a fruitful direction for Jesse to take his research? Allie's comments. Allie is a grad student working in Jesse Bloom's lab. We also had Tyler Starr, a postdoc, in the lab on that episode, TWiEVO 57. Allie's comment about antibody studies also got me thinking. Here, they have a pretty comprehensive list of which mutations afford good enough binding. In the interest of precluding all of the possible escape mutations, one would want to block all of them. Of course, money is always an issue, and some antibodies will be better at blocking several mutations than others. How would one approach a problem where one is looking for antibodies that are good at blocking multiple mutations? thanks for all that you do, Dr. Dave Jackson. Okay, so a couple of issues, or a couple of interesting questions here. So this notion of, so first of all, the origin, and sort of connecting to some of our conversation already. Is this from bats? And if it is from bats, in particular, why the Chinese bats? Why is this sort of a hotbed for coronaviruses? And so I think getting to that first question, so I think there is an important point here. And actually, I think Eddie Holmes, again, this evolutionary virologist headquartered in Australia, who has some collaborations with and has been involved in some of the sampling, has spoken about this as well, and can probably do so, or I'm convinced can do so more eloquently than I can. But yeah, so there is a good reason to step back and think about not just sort of one transmission event and sort of hypothetically. We'll talk a little bit about pangolins here in a moment. But certainly, yeah, bats have emerged as, I think, a pretty convincing reservoir of coronaviruses and the potential for them to spill over. And it is true. And this is sort of a theme that we've been hitting on, I think, in the last few episodes. And I think Simon Anthony made the point as well, that as you add more sampling of coronaviruses, that you start to change your perspective about the relationships between them. And so, and especially if you do this in a phylogenetic way, right? If you build these evolutionary trees. And so there, a couple of points. I think one is to remember that any evolutionary tree is a hypothesis, right? So it's based on the samples that you have, the alignments that you built, and then the algorithms that you use to try to discern a relationship between them. In this case, the relatedness of the virus strains. And so one thing that we really need to keep our eye on as more sampling is happening is, how good are these hypotheses that are being built? So how complete is the sampling? We're just, barely, I think we have we're undersampled so far, and vastly so. I think Simon made that point as well in last episode. And then, we've talked about this a little bit too, which is, so a lot of the alignments are based on, differences in point mutations. When we start adding more complicated things, recombination events, indels, the trees can be scrambled depending on what part of the genome you use to assemble it. Turns out just using an entire genome also has some pitfalls to it. And so I'm kind of going a little bit on a tangent here, but I did want to make this point is, so how do we try to test the veracity or the robustness of the hypothesis? When you look at a tree and the relationship between viruses, and you try to correlate that with a geography or a host where it was sampled from, you often see these numbers on the tree, the so-called bootstrapping numbers. And so what are those? So this is an attempt to just take a subsampling of your data and then assemble a tree to see how robust those relationships are. And so if you saw the number 75 on a tree that took a virus that was sampled from a pangolin, from a virus that was sampled from a bat. And what that means is if you draw that tree 100 times, basically 75 times it will look like you see it. 25 times that relationship will be different. And so to remember, even as we're both undersampled, creating hypotheses that could, that need to be kind of, I think, held in that sort of confidence. Like we're really trying our best to infer some things that are pretty complicated. And so anyway, just sort of, I think, a couple of more general points along those lines. As for the, is there something special about the Chinese bats? I guess my intuition, I'd be really curious what you think as well, Vincent, but my intuition is that they're kind of these microbial almost contingencies of history where it just turns out that through your private history of exposure to infectious microbes, that this will, in the very long run, impact the immune systems of the populations that these things are replicating in or sampling in. And so it's almost like the past, interactions that you've had will leave you either resistant or susceptible to the viruses of today. And so through that sort of long view of evolutionary history, I think you can imagine that just as these viruses and other infectious microbes were kind of sampling out in restricted geographies, that you just end up with species of bats in this case that have different immune systems. And in fact, we see massively strong evidence of that, that if you start to look at the phylogenetics of the relationship between bats based on immune genes, that actually the branches of the trees can scramble because evolution has been moving so quickly. This is sort of the notion of the Red Queen, or the recurrent genetic conflict. Almost feels like an arms race to kind of oversimplify it. So anyway, you end up with all these diverse immune systems that then will be either vulnerable or susceptible to the next thing, in this case, modern coronaviruses. And for whatever reason, there's a preponderance of these viruses showing up in Chinese bats. But remember, we also see corona-like viruses, and we talked about this quite a bit at length on the last episode, TWiEVO 58, entitled "Not All Coronaviruses ACE2 the Test." I have to just say it, not too shabby of a title. Anyway, and certainly we see on that tree some added bats from Africa, for example, some added bats from Bulgaria. And so the coronaviruses are in bats. I think we're just glimpsing at that. They may not be as prevalent in the bats of Seattle, or the bats in Texas, or other places in the US or South America. That doesn't mean that there aren't other things kind of percolating or smoldering under the surface. And as there's more human to bat exposure, human to wildlife exposure, generally speaking, that's where we start to almost ask for evolution to do these natural experiments to test whether there's spillover potential.
I don't think it's just China bats. If you look at the 7 coronaviruses, right, 2 of them originated from bats in China, SARS and SARS-2. MERS coronavirus, probably bats elsewhere, bats to camels some time ago. I don't know where those bats were. And then the 4 common cold coronas, 2 of those originated in bats, 2 in rodents. We don't know where that happened. So I just think recent history puts these 2 near us temporally, and so we think China, but I don't. As Simon said, bats everywhere have coronaviruses and other viruses. So I think not.
Yeah, I agree. And that one of those common cold strains, OC43, cows, I believe, in North America was the proposed spillover scenario.
I think that cow was the intermediary, yeah. So they all have intermediary hosts, and yeah, cows are one of them, yeah. OC supposedly stands for old cow, huh? Jordan writes, TWiEVO, you're discussing possible mechanisms for recombination in coronaviruses. There's evidence that the exoribonuclease NSP14 mediates recombination. There's a bioarchive paper by Mark Dennison that gives good evidence that this is the case. And Jordan is a PhD in medicinal chemistry. Now, you say, Nels, we actually discussed this paper?
Yeah, great call, Jordan. We have similar tastes in coronavirus preprint. So this is episode 55 of TWiEVO, Coronavirus Evolution from Soup to Nuts.
Okay.
And we picked this up, and it was exactly on this theme of, as we kind of think about mutational mechanisms of mutation, to broaden our horizons a bit, to not just think about the point mutation, the letter change, whether it's DNA or RNA, but the notion that long stretches of nucleic acids can recombine, and viruses are particularly good at this. Coronaviruses also particularly good at this. All kinds of mechanisms. Flu virus, you have reassortment, ways of kind of scrambling the deck, and to mutate and adapt, or have the fuel for adaptation, basically.
Okay.
So Jordan, check out TWiEVO 55, where we discuss this at length. And here, a really cool kind of mix of some emerging sequencing, long-read sequencing technology to try to see what's going on. So not only basically the full quote unquote viable replicating genomes, but the idea that coronaviruses and other viruses are spitting out defective genomes constantly that might be products of recombination. And although these will never go on to replicate on their own, that they might provide sort of more fuel for that fire of recombination. If you have these little fragments, and there's more diversity among them, that then this could sort of contribute to diversifying. Another note of caution real quickly here is the technical artifacts that can come from long read sequencing, or and this is what the Dennison lab spent some time doing, working in collaboration with Andrew Ruth's lab, to try to get really high quality, or to feel like they have confidence that they're finding things that are really in that sample. They're not just sort of built as artifacts. We'll see. So I'm guessing that I think that came out in April. I think it's under peer review. We'll see if it comes out after peer review, they kick the tires on that, some of the technical details, and if some of those results hold up. But really, yeah, I think a really important and cool preprint.
Cool. All right. One more, Nels.
One more. We're almost emptying out the mailbag. Feels like a relief. This has just been kind of sitting on my shoulders, Vincent. all these great letters for us to kick around and to get on the record. And so here's the last one. Tom writes, holy mother load of cool stuff. This paper is a portal into a lab where two lead scientists' brains share a lockdown lab time and spew onto the surface of innocent yeast to shed light on possible CoV-2 entry mechanisms and their real-time evolutionary processes. Good Lord, so cool. My favorite part of TWiEVO was the last bit where Tyler and Allie, so this again is our episode, this was with Jesse Bloom 2 episodes ago, I guess, TWiEVO 57 with the Bloom Lab. So my favorite part of this TWiEVO was the last bit when Tyler and Allie gave their what's next statements. And Tyler pointed to evolution, and Allie went towards immunological impacts in and around the receptor binding domains that this paper explores. And I speak for Twixers in general, I think, have been scienced. After a few hours of reading this paper, listening and re-listening to the show, I finally cracked a post-yeast elixir, which was embodied in a very cold porter. Thinking of the lack of blue and the otherwise sea of red from figure 3b showing mutation effects on binding, and the most satisfying and suggestive models in figure 7, mutational contrast of antibody epitopes. Oh, and actually this point came up in that last letter. I don't think we quite tackled that. So this notion of escape mutations or surface, where the mutations are happening. So that blue and red sea, this is the crystal structure of the spike, and then where mutations are accumulating that actually are viable and could be involved in escape. And so it's a story both of conservation, that sea of red, or I guess sorry, I'd have to look back. I guess it's probably the blue, and then the red are the places, the hot spots of change would be my best guess there. Okay, so sharing this data in the Altair, interactive statistical visualizations for Python that came out recently, calling out Sarah Hilton's work in the paper that was referenced by Vanderplas and others. This was a well-spent hour or two. Thanks. I hope to come back with this with some refined eyes after further learning and reading spawned by this thing we are all experiencing. All right. Well, Tom, thanks first for your enthusiasm there. And I'm glad that you kind of crossed over from both thinking about yeast display of these mutants to also drinking a cold porter for your efforts there. And so, and this was sort of, one of the take homes or one of the main points for why did they go through the trouble of making something like 4,000 mutations, and then having the ability to start talking through how, both natural antibodies or sort of related interventions, as we're thinking about vaccinations, might have durability or not based on what's sort of the mutational playground that this protein might sort of traverse, or where things are conserved versus sort of changing the most rapidly. And again, it's a single protein, a few thousand mutations, but the biology is so complicated, right? So are these mutations, or some of the mutations, could they be involved in ACE2 recognition, the receptor recognition to get into the cell, depending on the host? Could some of these be, selection of immune systems imposing on this, so that you have to have these, if you have these mutations, you'll be sort of vulnerable to a very rapid immune response, which will dampen the ability of the virus to replicate and move on to a new host. And so it's sort of this whole combination of things. So in terms of, and this is kind of also circling back to the last letter on this idea of, should we raise antibodies against every single variant out there? I think the answer by and large is actually no. And I think Jesse, really illustrated this well when he spoke about how, a lot of the mutational space that you sample probably doesn't have a lot of functional impact. And in fact, there's some reasons for optimism that, some of the epitopes that are being used to raise an antibody response as part of a vaccination or a related sort of medical intervention, that these have the likelihood to be somewhat durable. that you're not going to just sort of hit a point mutation that's going to all of a sudden, you're going to have this massive escape where all of a sudden all of this work that's been going in over the course of the last 6 months or so, sort of this hyper pace, will go out the window. So yeah, it's an important, sort of, I think, to how do you calibrate between, the ability to sort of quickly see and describe all these mutations to what is sort of the real life potential outcome, and sort of kind of have that balance, which is something that we're all still working out and trying to gain a handle of. But I think in general, that if sort of history holds, if our experience with epitopes like this and other sort of vaccine strategies, et cetera, most of what the deep mutational scanning has sampled is not going to be functionally consequential.
All right. Very good.
All right. Thanks, everyone. So the mailbag is cleared.
Fill it up, folks.
Fill it up. We'd love to hear from you. Kick around some of these ideas. So, Vincent, we're about an hour, a little more in, but I do want to just spend maybe 5-10 minutes on this paper. This will wrap our last loose thread.
Let's do it.
The fifth year, coming up on our 5th year anniversary. So, in fact, this was before I think in January, before the pandemic, the SARS-2 pandemic really kind of changed everything, and including the attention that we've placed to this across as you've seen, across the This Week in podcast empire, the Twix empire.
Listen to that. We've done TWiEVO, TWiM, Twin, and did I say TWiM? Twin.
Yep.
Yeah, the only one where we haven't really talked about at length is TWiP, I guess.
With Dixon, parasitology.
Yeah, all the others we've touched on it in some depth. So it's really changed the world. It's changed the microbe TV world.
That's for sure. I would say it's changed the entire world too. So anyway, before we were kind of our attention, for very obvious reasons, was drawn to thinking about coronavirus evolution, there was this great preprint that caught my eye. And this has been sort of sitting in our show notes, just waiting for an opening. And so I'm going to take it here. So this is a mysterious 80 nanometer amoeba virus with a near one complete ORFan genome, challenges the classification of DNA viruses. And so, there's more viruses in the sea, so to speak. This is an aquatic virus. It's not an ocean-going virus, but I think they got it from virus prospecting in a muddy creek in an artificial lake in Belo Horizonte, Brazil. The lead author here, senior author, is Jonatas Abrahao. First author, Paulo Borato. And so, a really fun kind of creative crew. So basically, you take amoeba, in this case, it's Acanthamoeba castellani, which is a well-known laboratory critter, a model system among the amoeba that's been domesticated in many labs. And using this amoeba as sort of a canary in the coal mine, or in this case, an amoeba in the water sample, put them into like 96 well plates, some cases thousands of them, and then expose them to this muddy creek water. And basically, some of the amoeba get sick. And so what that means for a single cell amoeba is that they lyse. And so around 3 days, you could just look at the bottom of the plate, and in each of the wells, the amoeba will be replicating. It will be kind of turbid. It'll be cloudy. But if they're sick and they lyse, now you have a clear well. And so there you go back and you ask, what is it that caused those amoeba to lyse to explode? And so they do that for a few. They actually use a pretty powerful microscope using cryo-EM, electron microscopy, to be able to go all the way down to see something like a virus particle. And indeed, they found some virus-like particles. They were able to recover the DNA, in this case, the double-stranded DNA, from these viruses. And they discovered an entirely new virus to science, to the world, which is about 50 kilobases, has 74 predicted genes. And the way you do these predictions is you look for a start codon. So this is in the genetic code, the message or the the code to start translating a protein, followed by an open reading frame before you hit a stop codon to stop translating the protein. And what's wild here is that when you do that, among the seven, there's more sophisticated ways to make these predictions and more variables you take into account. But when you do that, of the 74 predicted genes, none of them match, or I guess 74 out of, I think that might be 90% of the genes, none of them match any sequences searches with the genetic databases that are out there. And just to give you a little bit of context here, or calibration, the genetic databases that we have today are pretty massive. I mean, all the way from the human genome to all of the coronaviruses, everything in between. And the fact that you can't get a single match for 74 genes really says to you, what's going on? We are, we might puff up our chest and say we have this massive repository of genetic data, but we're really at the infancy in terms of knowing what's out there. Or we've discovered this crazy thing that no one has really ever seen before.
So now they have one protein with a clear ATPase motif. So in fact, while there may not be any homologs, they definitely have motifs that are known, right?
Absolutely. So it doesn't drop from like outer space or something like that. An
ATPase would be part of some protein that requires energy, right?
Correct. Exactly. And so, viruses are manipulating or influencing how energy is used in the process of replication in the context of a host cell.
I'm surprised they didn't find any, polymerase motifs, because that's very conserved, as you know.
Oh yeah, no, exactly. And so that's kind of what's exciting about this, right? When you get these like total outliers. And so, you need to ask a few things. Are you really assembling the whole genome?
Mm-hmm.
Or for technical reasons, are you missing something that would kind of put you on that footing to see some of the core machinery that sort of, reflect most of the virology that we're familiar with? Or is this really a different strategy for viruses to replicate? And so that's sort of the case that they're making here.
Yeah.
In the end, I think they conclude that 90% of the gene sequences had never been observed before. So in that 10%, and I don't remember all the details, there is at least a little bit of a foothold to, to kind of quote unquote traditional virology, in addition to the cryo-EM, in addition to the very clear sort of morphology as well.
So when they put this on their phylogenetic tree, where does it fall? Is it an outlier on its own?
Yeah, good question. So let's see. It's been a while. We're just kind of dusting this off from the show notes here. I did see, so I'll stall on that. It's a great question. I'll stall while I'm kind of looking through to just say that the paper was just published, actually. So it was a bioRxiv, I think was posted maybe sometime in January or late last year. And then just showed up a couple weeks ago, I think in mid-July, in PNAS, the Proceedings of the National Academy of Sciences. And so actually the paper, the preprint went through peer review and then got published in the time it took us to actually identify it to then to report it here, to share it on TWiEVO. So yeah, so there is a tree here, and it's not all by itself. There is a branch to Acropora millipora.
Yeah. I think this is based on a few proteins where they could see motifs, right?
That's right. Yeah. And so that, that's not a virus. I think that's a host protein that they're using here. And so a lot of times these viruses, especially these sort of large double-strand DNA viruses, they're gene thieves, they're gene hunters. They somehow acquire by horizontal transfer genes from their hosts. And so, so there is that link in actually, between hosts in some cases, or like, organisms or species that are in co-infection scenarios in the open reading frames you find. So there's also tRNAs encoded in the genome. That's another example of this. tRNAs involved in the translation process of the host cell. And so, these viruses are manipulating the way your core cell machinery works to become these virus factories in a sense. And so that does give you, again, some footholds onto sort of where they're coming from and how they're operating.
So they have one protein which puts it in the Marseille virus on a phylogenetic tree because of that one protein. And then they have another which puts it with a eukaryotic gene, as you say, gene 69, which is a bifunctional DNA primase polymerase. It clusters with eukaryotic genes. So very, very much like other viruses, it's kind of a hybrid with viral genes and cellular genes.
No, exactly. And so that allows, so then it raises really interesting questions about the origins. So it could be, so I'm kind of setting this up in a sense that, did this thing just drop from outer space, or it's some completely new thing? In fact, it might trace its viral ancestry back to some classes or, the viruses, et cetera, that we know quite a bit about. And then it's just sort of shuffled the deck through this kind of hybridization of borrowing or stealing genes from different sources along that evolutionary course. And then you end up with something that's quite different. And so that's where, you might give up on a sort of viral polymerase to grab one from somehow a host polymerase might take over. And so that's I think one of the fun sort of points here is to recognize how promiscuous these viruses are in diversifying. And they can kind of grab parts lists from hosts or co-associated organisms or other viruses that they might be found in sort of these complicated co-infection scenarios.
So that's why they call in the title that the virus challenges the classification of DNA viruses because it doesn't really fit in with anything, right?
Yeah, exactly.
Well, it tells me that there's a lot more virus space out there that we haven't explored, right?
No, that's right. And so, and this theme kind of keeps repeating. So maybe a decade or 15 years ago, some of the very first quote unquote giant viruses that came online, the Marseille virus class is an example of this, where these French scientists were just messing around in like water cooler towers, filter samples. And then, the breakthrough was instead of filtering out the bacteria, where you're throwing away these giant viruses because you're filtering them out, to actually include it all. And then all of a sudden there's this like hiding right in front of us, or these like bacteria-sized viruses with these massive genomes. And so, it's kind of, and the first time blast searches were done or database searches were done, some of those sequences are very similar, like 90% don't match anything. And yet, here we are 15 years later, kind of repeating, where we're just seeing similar sort of, very little connection to anything we've seen before.
So these sequences go in the database and then they could be hit in a future blast, right? And then it won't, some other virus will go, oh, look, these genes are homologous. I'd really be interested to see what Eugene Koonin thinks of this, because he often had, so this is a group in, as you said, in Brazil, right? And also they collaborate with the Marseille group, but Koonin often has his own opinions. In fact, a number of these individuals were at the giant virus meeting last year that we went to.
That's right. That was really fun. You, Rich, and I were there.
Good.
Twixers. So
maybe by the next one we can travel again, right?
I hope so. Man, I hope so. That would be great. So just to kind of wrap this one up, so they name it, discovering something that's only 10%, like a little trace of homology to anything you've seen, you get to name it, right? So they named this Yara virus braziliensis as a tribute to an important character, Yara, the mother of waters. Remember, this is from a muddy creek, of the mythological stories of the Tupi Guarani indigenous tribes. So a really cool local connection from where the virus was discovered.
Nice. Very nice.
Yeah.
All right. Can't believe we finally got that.
Did it? Whew, I can't.
After so many months. Cool.
Just great. I'm just feeling like a relief to kind of go through our summer cleaning here from our TWiEVO show notes. So let's wrap it up, Vincent, with some science picks.
Okay.
I'll go first. So I wanted to highlight a blog from Ambika Kamath. This is a postdoc, or a super postdoc, actually. So she's a behavior ecologist. She works on how animals move through space and its impact on evolution. And the animals she thinks about are lizards and spiders. She's currently a Miller Fellow. This is sort of like a super postdoc at California, Berkeley. And she's going to open her lab soon at Boulder, University of Colorado, Boulder. So if you're looking for a cool topic, take a look at the burgeoning Kamath lab. But the reason I wanted to specifically highlight her blog was this series she's done called Doing the Work. And this, it's a series of at least 3 blog posts that relates to the sometimes, in this case, awkward conversations you might have, she's having with her PhD advisor, really well-known lizard ecologist, Jonathan Losos, who was at WashU, then was at Harvard. I think he's back at WashU now. But conversations that I think many of us are having in our labs or in our communities about issues of diversity, equality, and inclusion. So, and Vincent, we just saw even in the last couple days here, some of the Black Lives Matter related stuff with this really awful story unfolding in Kenosha, Wisconsin. Some more police violence, and then the rippling repercussions of that. And so, with the George Floyd situation from earlier this spring, this really was an eye opener. And certainly, I think this is echoing through science communities, through labs. It certainly is in mine. And so I found this blog. In my lab, we've kind of teamed up with another lab, and we have meetings every month or so just to discuss these issues, diversity, equality, inclusion, and to try to educate and to move forward in a way that's more positive. And and you don't have to look far into the news headlines to keep feeling like we're still have a ways to go and that progress is slow. So anyway, I wanted to lift up this blog and also maybe a quick advertisement for the Cometh Lab as a interesting evolutionary ecology place to check out.
I like blogs very much. I think it's wonderful to write and put it out there. I wish more people would read them.
Yeah, agree. How about you, Vincent? What's your science pick of the week?
Well, I have a page I just put together called TWiEVverse. TWiEV. That's TWiEVverse, like verse, poems, and so people have been sending in haiku, limericks, poems to TWiEV. So I decided to start collecting them on a page.
Yeah, good idea.
And we have a bunch here. Most of them are pandemic inspired, but we used to run actually verse contests, haiku or limerick contests, and so one of them is left over from that. But people are very creative, so I appreciate it. I just, after the last one, I said, let me collect them. So I've been trying to find all of them. And if you're a TWiEV listener and you sent one in and you don't see it there, let me know.
Yeah,
but they're very good. Really, really good stuff. Thank you all.
Great creativity. This old saying that science is really more of an art than a science. I don't know if I go quite that far, but it doesn't hurt. It certainly helps. And this is a great example of it.
So it looks, we're back to our picks now. So if you want to send in a listener pick, you can do that. TWiEVO@microbe.tv. That's where you'd also send emails or comments or questions and so forth. We'd love to hear them.
Yeah,
and of course, all this stuff resides at microbe.tv/TWiEVO. If you like what we do, consider supporting us, microbe.tv/contribute. You can find Nels Eldy. He's at cellvolution.org, and on Twitter, he's L Earlybird. Thanks, Nels.
Thank you, Vincent. Appreciate you taking some time from your working vacation to catch up and go through the mailbag here. And we'll have a lot more to say about this the next time, but it's been a great 5 years. And
you bet.
Next
5, right?
Yeah, now we're talking.
I'm Vincent Racaniello. You can find me at virology.blog. The music on TWiEVO is by Trampled by Turtles. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, stay curious.